Hello, welcome to the video. In this video I'm going to be explaining factors that affect enzyme activity. Now enzymes are biological catalysts that I mentioned last video, and they are made of protein, which I didn't actually touch on. Now proteins can have their structure changed, and we're going to look at what might actually affect them now. Now there are two factors we need to know that affect enzyme activity: temperature and pH. I'll start off with temperature. So, we have another graph that we need to look at. This one is showing temperature on the bottom and enzyme activity on the side. Enzyme activity just means how well the enzyme works and how much it can speed up our chemical reactions. Now, as we increase the temperature, the rate of the enzyme activity also increases. Now, it increases up to a point which we call the optimum temperature. Okay, so here would be the optimum temperature. So there's optimum temp, and we can work out the optimum temperature by simply drawing a line down to the x-axis and finding what the temperature would be. Now in a human being, that would be 37 degrees, because that is the temperature at which we function. That's our body temperature. Okay? So that is enzyme activity. Now the reason it goes down is because after that point, the enzyme structure is changed because bonds are broken within the enzyme. And we call this denature. So the enzyme has broken down, changed shape. Now, think about the last video I posted about enzyme structure and function. We know that the enzyme has to be a specific shape to fit its substrate. So if the enzyme denatures, the active site will change shape, and the substrate, the molecule it's catalyzing, can no longer fit, therefore the reaction will no longer happen. So that's why temperature affects it. We need to know how to explain it like I just explained. The other factor is pH. Now pH is how acid or alkali something is. Okay, so at this end, on our pH scale, acid is 1, alkali will be 14, and neutral is 7. Okay, so that is the pH scale, and obviously as we go this way it gets more acidic, as we go this way it gets more alkali. Okay? And this can also affect enzyme activity in the same way. So, just like so. So the pH, as the pH increases, the enzyme will function best at different pHs. So if I look at it here, this would be my optimum. So that means it would be the best pH that it would work at. Now, different enzymes have different optimum pHs. So an enzyme that worked in my small intestine would have an optimum of about 7, so around neutral. Okay? But an enzyme that worked in my stomach, obviously it's very acidic in the stomach, would have a much lower optimum pH. So it may have a graph that should look like this and function best around 2. Okay? So that is temperature and pH. They can both affect the shape of enzymes, therefore affect the rate of enzyme activity. You need to be aware that different enzymes may function best at different pHs, and understand that temperature and pH can cause the enzyme to denature, change shape, and stop it functioning. Okay? Thank you very much for watching. Please like, share and subscribe. If you have any requests, please leave them in the comments. Thank you very much. Follow me on Twitter at Rushcliffe Bio.